Good day. My name is Grant Warnick. I'm the lead research scientist at Quantum Genetics, and today I'd like to discuss leptin genotypes and cattle production. At Quantum, we get a lot of cattle producers sending in samples to get genotyping done for leptin, and we often issue results in terms of leptin TT or leptin CC. And I'd like to just take a couple moments to discuss to sketch out what is leptin and how it is related to cattle production. So we'll just say leptin is a hormone, and it is released by mammals' fat cells, and it goes into the brain and it initiates the feeling full signaling cascade that we get when we eat a meal and we start to feel full. That is a hormone being released by our fat cells going to our brain and beginning that process. What researchers have found a few decades ago when studying mutant strains of mice is they found a particular strain of mice that were extremely obese, and they knew that there was a mutation in them. There was a single gene mutation, and eventually they would name this gene leptin after the Greek term leptos, meaning thin. So if there's something broken in this gene, it resulted in obese mice. And so later on, during the genomics era, cattle producers were very interested to find out what are the genotypes of leptin, to see if there's any mutations in leptin, any particular gene signatures that may result in increased feed production or weight gain or something like that. And what researchers found was that if there was a C, there was a C in a particular position in leptin that had been transferred in some animals to a T. Now that potentially had some importance, because if you step back for a second and you remember that DNA is the genetic code for proteins. So the specific spelling of a protein requires the right sequence of letters. And now there's only 4 letters in the DNA language, if we just follow this analogy. So there's A's, C's, T's, and G's. And so the sequence of those A's, C's, T's, and G's and the length of the word results in the protein that is being produced. And so if you change the spelling of that or change the arrangement of it, you're going to impact the protein being produced. To just give you a quick example, think about if you're going to spell cat, C-A-T. If you were to change the C to an H, you'd change the meaning. So in this case, with leptin, they have a particular sequence of A's, C's, T's, and G's, and at this one location, there's a C that gets transferred to a T. Now, when they followed the T animals, they found that they gained weight a lot faster than the C animals. And of course, when cattle are being bred, they inherit one copy of leptin from their mom and one copy of their leptin from their dad. And if that leptin on the mom is a T and the leptin from the dad is a T, that animal is a TT, leptin TT. Or if it's a C from mom and a C from dad, it's a leptin CC. Or if it's a T from the mom and a C from the dad, it's a leptin CT. That's what we call it. And so when they followed these animals, they found that the CTs tended to be the smallest, they found that the TTs tended to be the heaviest, and the CTs were in the middle. And so that's the relationship. That's leptin TTs and CCs and CTs, and that is the relationship between that genotype and cattle production. We'll end it there. Thank you for watching. Please tune in again for more information about using cattle genetics and plant genetics on food production. Thanks for watching.